We're going to be covering the overview of drug discovery. We're going to talk about what is a drug, what are some pathways involved, what are we talking about when we say bringing a drug from concept to clinic or concept to consumption. So I talked about myself already. Now we're going to talk about the topic. So what's a drug? The complexity of drug discovery process, of the whole process. It's long. It's a 7 to 15 year process before the COVID vaccine. Now it became a year process. But in general, and that was an emergency scenario. In general, for a normal therapeutic, whether it be a cancer therapeutic or a normal over-the-counter therapeutic, it's a long process, 7 to 15 years. And think about, well, if you're on, a lot of us are kind of set in where you want to be in this process, gene therapy. But if you're unsure, think about when we cover these different topics from the early drug discovery to clinical drug discovery to post-clinic life cycle management of the drug. Where do you fit in? And think about where do you see yourself 10 years from now, where you work. So can anyone name this molecule? Random question, but can anyone name this molecule? You drink it maybe every day.
Is it caffeine?
Yes, it's caffeine. Awesome, nice job. So caffeine is a drug. This is what most drugs look like. They are smaller molecules. Some of them are large. We'll talk about size. But a lot of them are less than 500 Daltons. Now, the reason why I showed caffeine is because once I talk to you more about my thesis, the compounds that I developed were derivatives of caffeine that actually caused cancer cells to go through a process called cellular senescence, which means they stop growing. So a drug could interfere with any pathway depending on the structure. The structure means everything. Now, here's an example of what we're going to see once we cover more into the different receptors and the different metabolic pathways that are involved to answer those first questions in drug discovery, which the first questions are: 1, which disease do we target? And 2, which pathway is involved in that disease? And also, which protein in that pathway or enzyme or receptor or gene is responsible for that disease, and we can either turn off or turn on more, hyperactivate, to cause a cellular response that is favorable for the diseased patient, meaning it mitigates the progress of the disease or cures it completely. So here's some examples of different cancer therapeutics that are involved in GI implicated cancer. So colorectal cancers, really colorectal cancers. And there are a lot of drugs. So these drugs that end in AB, those are antibody drugs. And then you have some that end in IB that are down here. These are inhibitors. And then you have STAT drugs, which are other inhibitors, but they're different structures. So the idea is that for a specific disease, which these are all GI implicated cancers, this pathway, then we have proliferative pathways, 2 of which are important, called the MAP kinase pathway, which is RAS, RAF, MEK, ERK, the one on the left in purple. And then we have the PI3K pathway, which is the right. They both do basically the same thing, which is to increase cell proliferation. Now, scientists and different pharmaceutical companies think, all right, we have this disease that we know from years of research is caused by the over amplification of this pathway. What are different points on this pathway that can be inhibited? Each one of these boxes is an enzyme. We have kinases, we have different phosphatases that are not shown here. We have other proteins, adapter proteins that are not shown. There's a lot here. And out of this entire pathway and all those proteins implicated, which one can properly bind a drug and inhibit it from its function? If we inhibit a drug in this pathway, it's like getting rid of a foot in the ladder. or getting rid of a step in the ladder. Yeah, not a foot. A step in the ladder. If you get rid of a step in the ladder, you're missing a step and you can't go on. So we can target, the idea is we can target multiple places in this pathway because these pathways are very intricate. There are still pathways, or there are still places and enzymes in every pathway that do not have therapeutics, that need therapeutics in order to cure that disease, or to cure it more effectively in combinatorial therapy, which we'll talk about again, or talk about later. Now, a lot of these drugs, you have a chemotherapy regimen. You have a regimen that is designed by doctors based on your individualized cancer mutations that is specific for not only blocking, let's say, RAF, this RAF1, but also block EGFR. It could block, and you can have 2 inhibitors being used because some cells might be resistant to the first one. So this is, we need to keep an eye out for drug interactions and combinatorial therapy uses of these different types of drugs. So the point is, for every pathway, there is many ways to block each step of the pathway. And that's something that we should keep in the forefront when we go over new pathways. So we're going to cover a lot of them in this class. We're going to cover receptor tyrosine kinases, which is this. We'll cover GPCRs. We'll cover a lot of other ones too, hormones, nuclear hormone receptor pathways, opiate receptor pathways, a bunch. We'll even cover cannabinoid receptor pathways. In all these pathways, there's multiple ways to block them. And on an exam, the kind of questions I'm going to give you for like the open-ended is name 2 ways or name 3 ways that you can inhibit this pathway that causes proliferation or causes a disease. If you block the, or how are ways you can do it? And I'll give you a diagram like this. So it's your job to see, all right, we can block EGFR, we can block RAS, we can block RAF, we can block MEK, we can block ERK. So yeah, just think about that. So there's a lot that goes on in drug discovery. So before we get in, that's more detail. But what is a drug? We'll start from the basics. So a drug is defined by any substance that brings a change in biological function through its chemical or physical action. So it should say chemical and physical action, because you're never going to have one without the other. So it's a molecule. Doesn't have to be big or small. Most of them are small. But it needs to change the biological outcome. So it changes a metabolic pathway, either on the protein level or the post-translational level, or on a genetic level, where it blocks a gene, or it does histone acetyltransferase, for example, something that changes the DNA replication, or changes the modifies transcription based on the DNA that's available from the histone. So all these drugs need to have, whatever drug we make, needs to have the appropriate size, electrical charge, shape, composition, properties, and metabolic profile. So we'll talk about all these in detail. The size, like I said, 90% of drugs that are in the clinic today are small molecules. And so when we talk about a field, small molecule drug discovery, which is what I'm in, is we mean by small molecule under $500. A new, well, not really new, there's been monoclonal antibodies around for a while, but it's an emerging field. Biologics are an emerging field that are going to have more automation. Synthesizing monoclonal antibodies is a popping. The field's popping. And we do have a couple of you guys who's mentioned that, at least one, that mentioned you're involved in monoclonal antibodies. So this is a field that I believe is going to be heavily explored more than it was in the past. And the reason why it wasn't explored in the past, because the clinical application for monoclonal antibodies based on a revenue perspective is limited, because antibodies are tough to store. They require a lot of money to produce. And they're proteins. Antibodies are proteins. So they require on a dry ice storage at least. to be active. But they're highly specific. You can make an antibody, and we use antibodies all the time in diagnostics. When we're detecting diseases or detecting or something in your blood or anything, we use antibodies and pull down assays to detect a lot of things out there. And they're very specific. So we have in Western blots, if you've ever done Western blots before, for example, in this protein, in this pathway here, this protein called ERK, ERK. There's a phosphorylated ERK, which means it's active, and a non-phosphorylated ERK, which means it's inactive. In a Western blot, we have antibodies that can actually bind to one versus the other. So one of them will not bind phosphorylated ERK. Well, actually, it would bind both. It will bind ERK and phosphorylated ERK. The other one will only bind phosphorylated ERK. There are tiny modifications in those antibodies that cause that difference in activity. Meanwhile, ERK is a huge protein, 30 or 40 kilodaltons. But this one phosphate changes the activity or the binding potency to an antibody drastically, reduces it to almost zero. That is how precise antibodies can be, which is why it's a great pharmaceutical tactic to drug discovery. So next, electrical charge. So electrical charge, shape, and composition are really all in the same boat, where we need a drug that binds to where it's supposed to bind. In order to do that, it needs to be a specific shape to fit the receptor. It needs to have specific charges. If it's charged positive at physiological pH, and there's a lysine in the way, it's not going to bind there because lysine is also positively charged. If there's an aspartic acid or glutamic acid, that's negatively charged at physiological pH. So it's more likely there's going to be a charge-charge interaction, and that positive charge on that drug is favorable. So we need to think about charge. Also, we need to think about atomic composition. So what atoms do we have? Do we have fluorines are usually good. Having too many nitrogens or oxygens are not that good because they can have metabolic activity, and the drug may not even get to its target. And that goes into number 5, properties of transportation from its site of administration to the site of action. So this means in the process of how it's administered, whether it's orally, intravenously, intermuscularly or intramuscular, transdermal patch, epidural, even rectal, whatever the case is, does it get to its site of action? And also, this is the metabolic profile. Is it inactivated to get there? So, for example, codeine, right? Well, yeah. So, any questions with this before we talk about codeine? Any questions with the traits of a drug?
What is a typical half-life of a drug like an average? Is it like 7 days, 24 hours?
That's a tough one. Within the hours, I say less than 24 hours. If I had to pick an average, less than 24 hours. Yeah,
Okay, so I know
most
like
drugs are
aspirin has a half-life of 7 days, and after my stroke, they told me I can only take it like once a week because it's not really necessary for a young person to take aspirin every day.
Yeah. Some drugs it's longer, but a lot of drugs it's honestly most of them are hours. Aspirin, because it's highly lipophilic, it stays in your tissues for a long time.
Okay.
Also, it has It has a lot of mechanisms. I mean, as a blood thinner, painkiller, and it has a lot of mechanisms, so it does stay in your system longer.
Okay.
But it really varies a lot. From most drug, if I had to put a label on it, most drugs are within a day, but some are longer than that.
Okay. I'm just
Good
curious.
question. Yeah, good question. Great question. So yeah, guys, if you have any questions like that, please ask. Ask. And another thing is, uh, a lot of you guys come from the genetic background. I'm not from in the genetic background exactly, so you may teach me something. You may ask questions that I don't know, and I'll look up and we'll talk about it and have a discussion and learn more together. So definitely ask questions like that. Okay, so let's go forward with prodrugs. Now, a prodrug is a type of drug, and what it is it's an inactive precursor that is absorbed or it is taken, and then it is converted by the biological metabolism into its active form. In essence, like the last bullet point sums it up, it's taking advantage of the natural metabolic processes to have a pharmaceutical effect. For example, codeine. Codeine itself is not active as, well, it is, but not as much as morphine. So if you take codeine as an injection, it's not going to nearly have enough as effect as if you take morphine as an injection. Because codeine, so as you take an injection, I didn't mention this before, but if you take a intravenous, you are getting 0% metabolism, at least to begin with. So 100% of the drug is entering your circulation. It has the ability to get to your tissues and have an effect, and have a numbness, a painkiller effect, and neuron desensitization. If you take it orally, which codeine is usually given orally, you have metabolism. You have metabolism in your gut. Most of the metabolism is in your liver by the cytochrome P450 system. So this system has enzymes that can cleave drugs. In this case, we have first pass metabolism and second pass metabolism. And we'll talk about that when we talk about pharmacokinetics in another lecture. But we have dealkylation of codeine, which turns codeine to morphine. The only difference between these two structures is that we have the methyl, it's demethylated or dealkylated on the codeine. So I mean, codeine is also called methylmorphine because it has that methyl group on the hydroxyl instead of the hydroxyl. So making that metabolism, and their cytochrome P450 system in the liver causes that metabolism, causes that enzymatic reaction to go from codeine to morphine. Morphine enters your bloodstream and becomes the active drug. So you're taking, there's a lot of drugs like this that are prodrugs. So you're taking advantage of the natural metabolism to end up with the active drug. So there's limitations to this because sometimes with prodrugs, if you have liver failure, so that's why you notice on a lot of drug commercials, there's that when they talk fast at the end, where it's like a lot of times they say if you have liver failure or liver problems or kidney failure, right? You may have a problem taking this drug and you should not take the drug. That's because a lot of the metabolism of that drug is either done in the kidney or the liver. So, and also it may be a prodrug where the drug doesn't have any effect unless it's metabolized by the kidney or the liver. So that's why we got to keep in mind of the pharmacokinetics, pharmacokinetics, meaning what is the body doing to the drug, how it's metabolized, how can we take advantage of that metabolism, and how can we use it for a prolonged or a better therapeutic effect. Any questions on this?
Professor, so what is the benefit of ingesting an inactive drug and then using your body to activate it versus ingesting an active drug? Like, why do you need that methylation?
Right. So the methyl acts as a protecting group. So if you eat morphine, right, the amount that will enter your circulation is significantly less because what will happen is this hydroxyl group might get reduced, or it might have off-target effects. Mostly, well, it's a combination of the both. And they don't even know really. So it's really both. If they did a full analysis, I bet they would tell you that because of this hydroxyl group, it might make off-target effects with maybe your albumin or your plasma proteins in your blood. And if you make an interaction with your plasma protein in your blood, you won't have any tissue activity. You won't have any activity of the drug in your tissue. Or it might bind to a gut protein. In that case, it won't ever pass through your liver. It might also bind to not the opioid receptor. No, it won't get there. It won't get there. So the point is, it might be metabolized. It might have off-target effect. Well, really, the answer: off-target effects, or it might be metabolized before. So the methyl or any other group is a protecting group. That's the main answer. that make sense?
Thank you. Yeah.
Okay, cool. All right, any other questions? Okay, cool. So drug size, so we have very small drugs, like lithium is a drug used for a lot of different psychiatric diseases. Also, we have very large drugs. So monoclonal antibodies are huge proteins, sometimes up to 30 kilodalton proteins or more, 100 kilodalton proteins. But most are between 100 and 1,000. So the reason why we want to keep small molecule drugs below a certain mass is because of and reason why drug discovery was first originated with smaller molecules is because of their diffusion properties through the cell membrane and also through other compartments in the body, going from the gut to the liver. Taking anything orally, we usually would take small molecules orally, like aspirin or ibuprofen, things like that. And the reason is because of diffusion. So for large drugs that are either peptide drugs or antibodies, they're usually injected or administered directly to where they have their effect. So wherever it is, if it's a drug for the brain, it's usually injected into the brain directly. It may not be able to pass the blood-brain barrier, which is another animal in itself, but usually these larger proteins, which is another limitation, is their transportability throughout the body is limited. Also, there's a larger metabolism. The larger your molecule is, the more likely it has sites on it that can be metabolized, cleaved, or glycosylated, or sulfonated, and it could disrupt the structure. So for drug discovery in general, we try to keep to this, it's a loose rule, Lipinski's rule of 5, basically stating that in order to have optimal drug conditions where we're limiting metabolic activity by the body before it gets to our target site, we want to have these things. We want to have no more than 5 hydrogen bond donors, so NH or OH bonds. No more than 10 hydrogen bond acceptors, so nitrogens or oxygens total. A molecular mass of 500 or less, and a log P, meaning its lipophilicity level, log P, less than 5. But these are loose rules. And also, we don't have to memorize these rules. These are just for our knowledge. But know that drug size is important and why it's important. Okay, next, drug shape. The shape of a drug is very important because of the receptor that it's binding to, or the protein that it's binding to. So a lot of these drugs are chiral, meaning they have a carbon with 4 different groups attached on either side. And because of that, we have different enantiomers. We have enantiomeric pairs. Sometimes different enantiomeric pairs of the same exact structure, or enantiomers, may have different activity, or completely, usually it's not completely, one's 100% active and one's 100% inactive, but it's usually, there could be off targets, more side effects with one versus the other, and also different outcomes. So a great example is the comparison between ephedrine and pseudoephedrine. So as we know, Sudafed, which is a precursor to methamphetamine. But the structure is very similar. The only difference is that the hydroxyl on ephedrine is coming towards you. Meanwhile, the pseudoephedrine is going away from you. That's the only difference. But ephedrine, just that little angle change of the hydroxyl, it is a beta and alpha adrenergic stimulant, meaning it activates the sympathetic nervous system, and it makes your heart rate increase, leads to vasodilation, pupil dilation, and other effects like that. So it could be in some pre-workouts. I don't read the labels for I don't take pre-workout, but ephedrine is a stimulant that's not as harmful. Like, you're not going to take adrenaline as a drug, but ephedrine is one of those stimulatory drugs. Pseudoephedrine, on the other hand, does have that activity. It does bind to alpha and beta adrenergic receptors, but it is a more indirect mechanism. It binds to those specific type of beta adrenergic receptors, mostly beta, that cause a increase in epinephrine, increase in the release of epinephrine, through an indirect mechanism, whereas ephedrine binds to alpha adrenergic receptors, which is in effect it binds to exactly where the same spot where adrenaline would bind or epinephrine would bind. But pseudoephedrine has this kind of dual mechanism, mostly indirect mechanism, where it causes the secretion of epinephrine, and this leads to mostly a nasal decongestion. It constricts blood vessels in the nose and really everywhere. It's a vasoconstrictor, and it's popularly used for respiratory infections and allergy medication. If you have a stuffy nose, take Sudafed. You'll be able to breathe better because it decreases the inflammation. So one bond difference can change the activity drastically. Any questions on that? What time is it? Oh, okay, we're good. So next, drug-body interactions. This is called, well, there's two types. We're going to talk about in this class pharmacodynamics versus pharmacokinetics. So pharmacodynamics is the action of the drug on the body. So we're talking about the chemical or the biochemical binding. Where is it binding? What proteins is it binding to? Is it binding off target? Is it causing another pathway to be activated or deactivated? What is the cellular response? And ultimately, what is the phenotypic response? There's been some drugs out there that we have little idea. This is crazy, but there are FDA approved drugs. And I'll try to find one sometime, but I know I've read them, where we know the phenotypic effect, obviously, because it cures the disease or helps a certain disease, but we don't exactly know the cellular response that's responsible for this phenotypic effect. So we don't know which pathways are being involved, but we know it does the job. An example I could think of that is being studied widely is any a cannabinoid product. So like any CBD oil or Delta 8, right? Or even THC. So these drugs, they have an effect of a calming effect. They bind to CB1 and CB2 receptors. We know about those receptors, but research is still being done on exactly what the cellular mechanisms are for those. And you have medical marijuana and other THC products that are clinically approved. And we know that they're safe. All we need to know for FDA approval is that they're safe, and the most important thing is that they're safe. The second most important thing is, do they complete the job? Do they have efficacy? But in that case, we may not even know the cellular activity in those cases, but we know what they do. So, and that's pharmacodynamics. Then pharmacokinetics, we're talking about the opposite of not what the drug does to the body, but what the body does to the drug. The main thing is ADME, and we're going to have to remember this saying, ADME. So absorption, distribution, metabolism, and elimination of the drug. So absorption, how is it being taken in? If you're eating it, if you're taking it orally, it's distributed. It's absorbed through your gut, goes through your liver, distributed. It's metabolized in the liver also. It's also distributed throughout the tissue. It's metabolized some more in the cells. There are proteins. There are protein channels. There are enzymes that can cleave drugs within the cell. There are enzymes like P-glycoprotein, which is a protein channel that acts to eliminate some drugs from the cell into the circulation again, and then the elimination of the drug through the renal system, or sometimes even through your lungs. So we don't think about alcohol as a drug. Alcohol is a very broad drug. It has a lot of ethanol, really has a lot of effects, has a lot of cellular effects because it's a really small molecule, and it has a lot of off-target effects. So yeah, if you get drunk, you may feel calmer, you may feel belligerent, you may have more energy, or you may feel calm and then your heart rate goes up, or you may have a combination of things. And this is really the toxicity of the ethanol that's causing that. But for the elimination, reason why I'm mentioning it is because it's a weird drug in terms of elimination. Obviously, it's eliminated through your fecal matter and through your urination and through your renal system, but it's also eliminated through your lungs. That's why when someone comes up to you and you've been drinking, they can smell it on your breath, because that is a way of the metabolizing of ethanol and the elimination of it. So not only the renal system is responsible for eliminating drugs. It really depends on which drug, and it depends on the really what we call volume of distribution, the volume of your body that the drug inhabits once it reaches your circulation, extent of absorption too. Okay, any questions on that? Okay, cool. All right, so the next part of class we're going to talk about, and we're really halfway done, we're going to talk about drug discovery. So what is the process, in depth, what's the process of drug discovery? So now that we know what a drug is and what to look out for in a drug, and of course, this is, of course, very introductory, what to look out for, we have our different properties, pharmacodynamics, pharmacokinetics. We're going to talk about the entire workflow of drug discovery. So I mentioned the first thing we want to do is, as executives of pharmaceutical companies, we want to identify a disease of interest. There's a lot that goes into this. We don't just look on the news. We don't just look at stats on the CDC website of diseases that people are dying from. We need to see, A, is the disease curable? Do we have an idea based on research of mechanisms or pathways that are involved in this disease? Does it cause a lot of discomfort in people? Does it cause death a lot? Because and also, how many people are dying from it? Those are all big questions that heads of pharmaceutical companies need to answer. There are some rare blood diseases out there. I can't even name them, they're so rare. That I'm sure we've all seen documentaries of little kids or other people that have these really rare diseases. There was this, I think one of these TLC shows or documentaries where there was this kid who was allergic to sunlight, and his skin was so sensitive that it would burn and hurt when it was sunlight. These diseases are very rare, 1 in 100,000, 1 in a million, 1 in 10 million. And even though there might be drugs out there, and there might be drugs that can be made to cure this disease, we don't do it. And the reason why is because of revenue. If there are 2 people, whether they be kids or adults, there's 2 people in the United States that have this rare blood disease. If it takes $1 billion to get this rare blood disease cured with FDA approval, with going through FDA approval, it's not worth it for companies to do that. That's the sad truth of it. So there has to be a lot of interest in that disease. So that's why we need to identify. That's why identifying the disease is a large part of it. And also, after we do that, we have to ask ourselves, all right, which protein target can we modify with small molecule drugs or biologics, large molecule drugs? Whether it be a kinase or a receptor. Usually we aim for receptors, extracellular receptors, because they're easier to go at. They're easier to attack. We don't need to go into the cell. Once we go into the cell, the chance of off-target effects are greatly increased. The effective concentration of the drug that reaches the intracellular material is decreased. So getting the drug to the receptor is one thing. Getting it through the cell membrane is another thing. We need to make a drug that's lipophilic enough to get across the membrane. That's a big deal as well, which we'll talk about when we go over pharmacokinetics more. So we have to ask ourselves that. Another important step, validate the target. Not only do I show you a diagram and say, oh, based on this diagram, that ERK, if we inhibit ERK, we inhibit the MAP kinase pathway, and we don't cause cancer anymore. Because we know that ERK is in this pathway. No, we need proof that turning ERK off artificially causes the disease to stop. causes the remission in cancer. The way we do that is a gene knockout. So what we would do is we would take a recombinant, or we could take a mutated gene. So we can either knock out the ERK gene completely, or we can make recombinant ERK that's all mutated, that will not bind to MEK. It's upstream effector protein. If we put this in mice, and we see, all right, the mouse that had cancer, which no longer has ERK in no longer has ERK in it. It has a RAS mutant, but no longer has MERK at all, or ERK at all. No longer has ERK. The mouse no longer has cancer. Therefore, if we inhibit ERK, we stop cancer. So that's how you figure it out. If the target, we call it target validation, that the target actually is implicated in regulating the disease. Then the next step, which this is where I come in, I would like to make these big decisions eventually. But then the next step is develop assays, usually biochemical, to inhibit the protein of interest. We go in the solution first, then we try to convert that to the cell. This is called target characterization and identification. So are there any questions on the first, and this is 1 to 3 years usually. Any questions on this? Okay. All right, next. So the next 1 to 2 years, sometimes more. It's probably more. Took me 3 to do mine, but I was only one person and with some help with my advisor. But anyway, so we need to develop an assay, which let's say we have an assay developed, a biochemical, like an enzymatic assay, that you can detect the amount of enzyme that's turned on or off by compounds, by molecules. All you need to do is put molecule X in there and see if it turns it on or turns it off. We need to identify a control that can inhibit and also activate protein activity, usually a positive or negative control. A lot of these controls are antibodies or peptides, molecules that will not be used in the clinical setting, but are tools. So they're tools for screening billions of compounds that are unknown, but we have a baseline. We have, okay, this signal, this flat line, usually it's a flat line, is what inhibition looks like. We need a compound out of our 200 or 2 million or 300 million for larger pharmaceutical companies, or even as small as my library that I tested, which was like 850. You need to see that, all right, if any of these 2 million look like the flat line, that is inhibition, and that's a hit. This is called high throughput screening. And that's why, so when and this goes into consideration when you make the assay. When you develop an assay development, there's a huge market for people doing assay development. And that's what I do. And there's jobs everywhere for assay development. That's why I was at my company for a little bit. I looked in the market, first job I could find, I applied, I interviewed, I got it. And if I didn't, I'd probably find another one. So there's a lot of jobs out there in either the biochemical or the biophysical or the cell-based assay realm of assay development. So when you're making these assays, you need to keep in mind how much material needs to be used. So can I do that? Can I do 2 million samples? What is the sample consumption? How much protein is it taking up? Because there's also protein purification biologists that make large batches of milligrams and grams of protein. Milligrams is considered a lot for protein. So you need to figure out how much consumption. If we have 2 million compounds, we better be running them in 1536 well plates. Not 96. If we're running at 96 well plates and we do 2 million compounds, that's going to take 6 months to a year on its own. And that would be a waste of material. So we need to go back to the drawing board and think of a better assay. We also, for 2 million compounds, need to have automation. There's a lot of automation involved when we're in high throughput screening. There's an instrument that at my current job called the Axis, which is a giant robotic instrument. Those of you who work in pharmaceuticals have probably seen these kind of instruments where they're robotics, and you put these plates on the robotics, and they move the plates over to the instrument, runs the plate, stacks it up, next plate, next plate, next plate. And these plates are pre-dispensed with certain compounds by an automated liquid handler, never by hand, because you have 2 million compounds that'll drive somebody crazy, or you need to give them an intense or an insane raise to make them do all that work. So there's a lot involved here. Then you need to confirm the activity. So out of those 2 million, you also have data analysis that's automated. So you write a script that gets and also this high throughput screening is done at 1 concentration. So we don't have a dose response curve yet. It's just 1 concentration, 20 micromolar, 50 micromolar, 100 micromolar. Not usually more than that, because then you might face solubility issues. Maybe 200, but never more than that. So let's say out of 100, these 2 million compounds at 100 micromolar, you can write a script or have the data exported in a certain way where it reports percent inhibition. And those of which who are over 50% inhibition, you confirm them as hits. Most of them, 95% or more, will be inactive because they are random compounds from a compound library that companies develop from old projects or from just having compounds and ordering like a baseline compound library that most of them are going to be non-hits to this protein. So after we confirm hits, we are then going to do more assay. We're going to do a dose response in the same assay to figure out an IC50, meaning the concentration that is responsible for 50% inhibition. And this will be covered all in the next couple lectures, but just to see the text of it. But this is basically what we do. And if we have an IC50 that's good, meaning low micromolar to high nanomolar, we want to take those, what we call lead hits, and develop them. So that's the next step, which is in vitro pharmacology, which is developing. And also, that reminds me, I forgot to mention, I had a little bit of Merck experience when I worked for Merck for a short time in their in vitro pharmacology department, which we did this. So we need to develop additional assays, otherwise known as, we call them orthogonal, meaning another thing that leads to the same conclusion of these compounds to validate the hit validation. This is important because sometimes in enzymatic assay, in every assay, there are limitations, and we'll learn about different assays. But in enzymatic assays, a lot of them have to do with fluorescence, TR fret, absorbance even, or other glow assay, ATPase assays, things like that have to do with light and color. Well, there's some compounds out there that have intrinsic color. And I've seen this in my own work, where you have a compound which, after it's purified and you put it in the solution, it's purple, or it's red, or it's brown. If you put that into the plate and you run it, it's going to interfere with the fluorescent signal, and it may cause a false response, either false activation or false inhibition, false positive or false negative. What you need to do is validate it in an orthogonal assay that does not have color as an effector. So if you have an enzymatic fluorescence assay, you may want to do SPR, which is a biophysical assay that does not involve color or not involve fluorescence at all. You may want to do a thermal shift assay. You may want to do circular dichroism. You may want to do ITC, which is isothermal calorimetry, DSC, things like that. And there's more. So, and then once we have these validated, we want to do structure activity analysis or structure activity relationship called SAR. So the SAR means, okay, now that we have a batch of 100, let's say we got this 100 down to 50, right? We have these 50 compounds. What is similar about all their structures and what is different? Oh, look, they all have the same bicyclic core. Oh, look, they all have the same amine that's that's 20 angstroms away from a carbonyl group. Oh, that might be important. So we do an SAR analysis of all of that. It takes scientists a few weeks to develop an SAR and then to get compounds and synthesize compounds that are similar and based on that data might be more active. And then, of course, we put this into the cell. And then this helps us determine the mechanism of action and where it's binding. So I know I just talked for a lot. I rambled. So any questions on that? And there might be. So yeah, ask away.
I have a question. So for this flowchart regarding how much you want us to know about it, do you want us to just have an idea of the sequence of events in drug discovery, or is it more in depth, like the description like of each step in the sequence here?
So more what happens after what. So you don't need to. I wouldn't say memorizing them. It wouldn't be to your benefit. Understanding it rather than memorizing. So if I ask you what makes sense to come first, developing a high throughput screening assay, or doing a cell-based assay, or synthesizing new compounds, what would you say makes more sense to do first? It'd be high throughput screening, because we don't know anything about. If you're doing it first, you don't know anything about the drug. I mean, the question would be better phrased than that, but you don't know anything about what you're looking for, so you screen as many compounds as possible. If you do a cell-based assay first with compounds, or you don't know what you're doing it with, you're wasting stuff. If you try to synthesize stuff, what are you synthesizing? You don't have any. There's no workflow there. So the logic of the workflow is what I'm more concerned about, if that makes sense. I hope that answers your question. Question?
Yes, maybe my question is not so relevant to this chart, but I was wondering if you could let me know in which step machine learning can help us to predict that drug, the structure of the drug. Is it in the HTS?
We can predict the mechanism of the drug. Is that what you said?
Yeah, you said about the libraries, and I think some I think instead of using high throughput screening, it can be used machine learning to predict the structure of that desirable drug or not. I'm not sure about
the. Of course, you're okay. So you're definitely right. Awesome. So this is a great point. That this is where this is a new approach where industry is going to high throughput screening is old school. We are now looking at computational. It's still in baby steps, or in the infant phase, we'll put it. But we've used this, and in most drug discovery projects now from big pharma, they're using computational chemistry, they're using computational biology, machine learning, where we can take structures of millions of compounds and we can fit them into in thousands of iterations, do an energy minimization model where we can minimize the binding energy or maximize the binding energy of a compound to an active site of a protein and find active sites. We can find different protein sites, and we can do this all through the computer. It does take a lot of time. High throughput screening takes longer, but it's also very expensive, and also it's difficult. But also there's a lot of skepticism about it, and people who I've talked to and I work with have skepticism about those techniques because we're at the point where it's getting better, but until you do the biochemical work, we don't know. If I had 2 million compounds and I had the access to do computational machine learning structure-based analysis, I would do that first. I might get the 2 million down to 500,000 or maybe 100,000. But until you do the biochemistry, you don't know. You don't get it down to 200 because the computer is the software isn't advanced enough to know, yep, 100% this one binds, this one doesn't. It will find ways for it to bind. A lot of them will come up as false positives, but it will definitely narrow down your search. So that is a great point that you mentioned.
Yeah, it's my research actually, because we want to engineer an enzyme, and we would have a lot of libraries of the gene. So we are supposed to work with HTS method for directed evolution, but recently I found a lot of scientists work on machine learning, and my question is that, is it supposed to replace HTS one day in future?
Yes. So in the future, that's what the goal is to well, really, the goal is to completely automate HTS if we do it at all, and to either make it completely in the computer, completely AI, or replace it with a dominant computational method. That's the future of drug discovery, but we're not there yet. But it's awesome that you're working on research like that. I'd love to hear more about it some other time. So that's awesome.
Thank you.
Yes, definitely an important point that I didn't mention. So to give you an example in real world, in my research, we did use, if you're familiar with Schrodinger suite, is a powerful software that can model proteins and optimize interactions with drugs. We used that to narrow down small library of about 1,000 to around 150. But then my hits were actually 3. So it does work to narrow it down, but narrowing it down to that smaller number, you still need to do the enzymatic work. But we're on the path to definitely on our way out with HTS, but it's still highly prevalent in the industry. Every company uses it because right now, look who's in these executive positions. People who've had clinical success with their drug projects from doing it the traditional way. Until we see real compelling evidence of the AI and machine learning techniques showing clinical proficiency, we're not going to see a full transition. But it's going to happen.
Yeah.
Okay.
Yeah.
So going forward, once we do our SAR, structure activity analysis, and we identify which parts and structural parts of the drug are responsible for binding, we need to identify lead compounds and focus on lead optimization of either 2 to 4 cores. This means, or even 1 core, really. Cores mean the cores of the structure, whether it's a bicyclic ring, tricyclic ring, some kind of imidazole, or whatever the core is. We want to focus on that and develop a series of compounds for each core. The reason why we want to do more than 1 core usually is because what if 1 core fails miserably and it gets metabolized, or it just doesn't reach its target by any administration method? Then we spent millions of dollars to this point and the project is done. So it's better to have a few cores and a few series of compounds ready for analysis and for optimization. So through synthesis, we optimize that lead and just organic synthesis. And then we come up with the in vitro pharmacology assays. So this is where we go from the optimized lead. Of course, we test it in the cell. We test it and make sure, all right, it has a 200 nanomolar IC50 by Western blot. That's really good for Western blot. Or it has a, whatever. And it has, in the enzyme, it has 1 nanomolar. In SPR, it has 100 or 500 picomolar. Very good. The next, we want to bring it into DMPK. So drug metabolism and pharmacokinetics assays. I haven't done these type of assays, but there's albumin assays to see protein binding in the blood. There's a plethora of assays to see, all right, what is the metabolism of this drug? Does it metabolize? And then if it passes those tests, we go into the in vivo models, the mouse models usually, maybe pig, but usually mouse models. And we see, all right, what is the phenotypic response? We look for safety and we look for phenotypic response. So does the drug complete its, or does the drug have any efficacy? And then we select a compound or a core for development. So any questions after this point? So this is where, after we're done with the model, the animal models, we have a drug ready for clinical trials.
Professor, I have a question regarding the first experimental step in deciding to discover a drug for a disease. Would that be to identify control compounds and inhibit protein activity, or to propose protein target to modify with a small molecule drug?
So the first steps, identify your disease and proposing a protein target, or really going from a metabolic pathway, the disease, the metabolic pathway associated with the disease, and the protein within that metabolic pathway, proving or validating that it is the reason for that disease to come, and inhibiting it will stop the disease. Target validation. So those are the first steps.
Thank you.
Yeah. Okay. So next, clinical trials. So I have not been involved in a clinical trial before, as a scientist, because my experience is in the preclinical drug development. But clinical trials, it requires a lot of people as well. So you could think, like, how many people are involved in drug discovery? That's a good question. And how do you fit in? There's a lot of people. There's thousands. Well, maybe 1,000 or so. But I think it's a homework question too. But that's more of a brainstorm question. You can look it up, because I don't really know the answer. It really depends. But you can look it up. So anyway, clinical trial phases. There are technically 4 phases, but we say there's 3. There's 3 before FDA approval. Phase 4 we consider to be after FDA approval, meaning post-market. So right now, for the COVID vaccine, we are on post-market, phase 4 analysis. There are side effects. There are conditions that people, there are accounts of, rare accounts of people developing blood clots, or heart murmurs, or heart or myocardias from, we think, is from the COVID vaccine. But it is very rare. But we're in that phase 4 clinical development, and we're in the longitudinal study. There has been, so a reason why people were a bit scared of the vaccine, or some scientists were skeptical even, were because we didn't have, it's not like a flu vaccine where we had 20 years of longitudinal proof that it was healthy. But a lot of these mRNA vaccines don't have any long-term effects. And as you can see, the COVID vaccine is pretty short-term, because you can get the vaccine, and now you have many people that were vaccinated 10 months ago who are now getting COVID again. So, but the point is that some drugs do have a lasting effect, and they might have 10 years down the road increase your risk for certain diseases. This is what the phase 4 longitudinal studies are for. And you see these commercials sometimes where if you take the drug X, if you took drug X for heart disease in the 1990s, you can be rewarded you could be part of this class action lawsuit against this pharmaceutical company because it causes an increased risk of cancer, right? That comes up, and that's always something that pharmaceutical companies have to keep in mind. But you can't dwell on that, because you're trying to save lives, and most of the drug administration saves lives, or greatly improves someone's life, or someone's treatment options for a certain illness. Those long-term effects, I mean, sometimes they're unavoidable. But anyway, to start from the beginning, phase 1 and phase 2 are basically the same thing. They have to do with safety. Phase 1 is safety, really only safety. And also we talk about, we learn the absorption and the metabolism and the side effects, otherwise known as safety, with 20 to 100 people. After this, we don't even care about the efficacy. These people might be healthy. So if it's a cancer drug and they're taking it, it's not going to have any efficacy because they don't have cancer. But if it's a hypertensive drug or a statin. Statins are used for heart disease if you have for high cholesterol. If you take a statin and you don't have high cholesterol, there could be some side effects. So you'd want to put it into people with high cholesterol or people that are at risk for heart disease that need a statin. So this 20 to 100 healthy volunteers, it depends what type of drug it is. So there's some drugs out there like some ADD medications, let's say, that will make a healthy person go crazy, but they're like Adderall, for example, right? If a healthy person, and people have done this in college, they take Adderall and they're healthy and they don't have a attention deficit disorder, and they take Adderall. Well, it makes you wired, and it's not safe. But if you need it for your attention, and you're not going to see as many side effects, you'll be more normal. I'm not going to use the word normal because no one is normal, but more attentive. But anyway, that's phase 1. Really targeting the safety and the side effects. Phase 2, you have more people, and you have a control group and a treatment group, meaning a placebo group and a treatment group. And usually with several hundred or even a couple thousand of the cancerous or unhealthy people that are suffering from the disease. And you want to test the effectiveness. And then and of course, safety. Also the dose range. This is something that comes into play for phase 2. Dose range, meaning we're going to see a dose response curves. We're going to see dose response curves later on in this class. Concentration versus effect versus dosage. Also, we're going to see TD50 curves, meaning toxicity curves. So how much of the drug can you take before you get toxic effects? For every single drug, the most harmless drug you could think of, if you take too much of it, an overdose, there is toxicity involved. You will have nausea, vomiting, headaches, or whatever it may be, or increased heart rate. You might pass out. You might have some other seizure, whatever. There's going to be effects. There's going to be side effects at higher concentrations. So we're testing for that in phase 2, and we're getting the idea of, all right, how high can we go in a patient before they feel nausea, vomiting? And that's where we put the low point on the curve. All right, that's the concentration which it starts at, where we have the nausea, vomiting, or at concentration 2 micrograms per milliliter, or that's low, but 2 grams per milliliter. No, that's high. 2 micrograms per milliliter of drug. They're going to be, or 20% of people will have nausea. Or at X concentration, 50% of people will. And you can make a curve out of that. And that's called a TD50 curve. Then you have phase 3. Phase 3 is where you take thousands of individuals who have this disorder and you test them for more, obviously, safety. You will see more uncommon side effects come into play. Like 1 out of 10,000 people will experience a blood clot, for example. Like that's going to happen here. Phase 2, you might not have seen that if you do a couple hundred, or you might have 1 out of 10,000 get a rash, a weird rash, or they lose their sense of smell for a week or something. So you may see this, and also. Yeah, that's about it. Yeah, and just, of course, further testing the efficacy. So in this entire drug discovery thing, our process, think about throughout this entire class where you fit into, if you fit into it at all. So for me, I like to explore. I like to get down and dirty with the science and think about the enzymes and the proteins and the pathways. And I've really found that to be my interest when I was younger, or in school, and I went with it, and I like that. But some of you might like the clinical aspect of it, where you're actually seeing the fruits of your labor. If I develop a drug, I'm going to be on the preclinical, the exploratory phase where I'm testing an SPR, not a final drug. It's a precursor to a precursor to a precursor of a drug class, even. So I'm going to be way down the line. But once this drug comes to fruition, I'll have a little bit of a pat myself on the back. but it's not going to be like somebody who was directing clinical trials, where they're going to be like, I brought this drug into the clinic, or someone who's an executive of all of the organic chemistry or the lead optimization process. Because doing the SAR is really drug discovery. I would say the nuts and bolts of it, I would say the foundation of drug discovery is the SAR. With everything else, you can treat people, you can do a high throughput screen, you do whatever you want. If you don't have really good scientists drilling into the SAR of, all right, which part of the compound causes this interaction or causes this enzymatic reaction or causes this effect. If you don't know that, you cannot have a drug. And you cannot optimize a drug to go from 10 micromolar to 10 nanomolar potency. It's all about the SAR. So if you get good at SAR, that's very valuable to any company. Okay, so like I mentioned, these drug discovery process, you're talking about millions of compounds. All the way, it doesn't say millions, but there's a step before here, it should be millions. All the way to one new drug. I mean, you might have 5 in clinical trials, more than one, or a few of the same class, a few of the same core, but you're going to have one drug that's going to have approval. And then you have this post-market surveillance, otherwise known as phase 4. So yeah, that's the whole process. Any questions on the process?
Professor, in the drugs
Yes.
discovery part, you mentioned that you modify with small molecule drug. So do these phases mostly apply to the small molecule world, or is this a slightly different route if you say you're doing biologics or gene therapy where
so
your screening is more expensive?
Yeah, so for gene therapy, I'm not really familiar with the screening, but it's similar. That's all I know. It's generally the same workflow, but instead of direct binding, you're looking for gene activity, I guess. But you probably know more about than I do. But for biologics, it's more of engineering because you can make an antibody perfect for an enzyme. You don't have a library of a billion or a million antibodies and say, oh, which one binds to this enzyme? You know the structure of the enzyme by an X-ray structure. You know the residues that can be involved in its inhibition. Let's say you do. If you don't, then yes, you need to do a high throughput screen of millions of antibodies or millions of compounds, which a structure that is like that compound can be put onto an antibody for that antibody to have that effect or that binding capacity, just like that compound did. So there's a lot of ways you can do it, but mostly for antibodies, they're engineered to where they bind perfectly to the enzyme to inhibit it. So it's modified. So this is traditional drug discovery, but this is usually 90% of the time this is how it goes. It's always changing.
Okay, thank you.
I hope that answered the question. Yep. Okay, so of course you want to think, where do you picture yourself in this process? And that's basically it for this week. And we went pretty long time. So what we're doing next week, we're going to talk about types of drugs: agonists, antagonists, inverse agonists, partial agonists, mixed agonists, drug targets, receptors, enzymes, things like that. The pharmacology